Here, question 2. Figure 2.1 is a diagram of part of a DNA molecule. Draw a circle on figure 2.1 to identify one pair of bases. It can be this one, it can be this one, it can be any visible pair, like a line in which the two nitrogenous bases are shown to be binded to each other. So you can write a circle around, draw a circle around any one of them. I chose to draw the circle here, which is okay. The percentage of T bases in DNA of a species is 29%. Okay. Calculate the percentage of bases that would be if base G in the DNA of the species space for working. This is basically for math skills, okay? It's not that related to biology. It comes more in paper too, but this is the first time to come here. So here, calculate the percentage of bases. So first of all, I need to know that A takes T and C takes G. What does that mean? That A usually bonds with T, like if this was taken to B, and this also, C to G. So when T is 29%, A must be 29%. And to know the base G, I need to know the remaining percentage, which would be the total of those added together. It's going to be 100% minus 29% times 2, which is going to be 100 times 100 minus 58, which will be 42. 42% remaining. They are divided equally, divided by 2, to C and G. Why? Because if I have 2G, it would belong to 2C. Each one G takes 1C. Just we use the same concept as we've dealt with A and T. So, C and G are both 42, then we divide by 2 to get that G has 21%. These are the steps for working. T is 29, and just the same as we explained. State the name given to a length of DNA that codes for a protein. This is gene. Explain how proteins are made by a cell. I can say that transaction happens and a copy of the gene in the DNA is formed, which is mRNA. You must mention the name. mRNA gets out of the nucleus through nuclear pores to be attached or pass through ribosomes in the cytoplasm. Here, this is the second part of our journey, which is translation. Translation occurs, and mRNA has a specific sequence of bases that codes for a specific sequence of amino acids. Ribosomes assemble amino acids into polypeptide chains. Which then fold to turn into a specific 3D shape. Here what happens. I have the nucleus, draw it a different color. Here I have, this is the nuclear membrane in which there are nuclear pores that we've been talking about before. Okay, here. And inside I have the nucleus. Okay. This nucleus contains DNA chain. I mean a DNA, DNA. And here this part is the gene that I need to copy that would be responsible for the protein that I'm going to synthesize. So this part, or this gene that I'm circling around, is going to unwind, and it's like, basically in slang language, it's going to detangle, then form the mRNA. The mRNA is the copy of this gene, so it's going to form and then get out of the nucleus through the nuclear pores. This is, imagine that the copy is going to form here. Drawing isn't clear. I don't know. Okay, I'm going to draw it again. And here, the lines. Okay, so this mRNA forms and get out of the nucleus through the nuclear pores. It's going to then go out, and we have like the cell. Like, here is the cell. Draw it back. Here is the rest of the cell, and I know that this cell is covered or filled, with this substance called cytoplasm. And in the cytoplasm there are little tiny ribosomes. But here I'm magnifying them so they can be big enough, because I'm here going to just zoom in to see the protein synthesis in a larger view. These are the ribosomes. So the mRNA is going to get attached to the ribosomes. It's going to be like this. And this mRNA is going to be like an instructor that's going to guide ribosomes in which sequence it would arrange the amino acids, meaning that it's going to just guide the ribosome to put, for instance, I have here these group of amino acids: the red, the purple, the green, the purple, the red, the yellow, the pink. Simply, mRNA can just tell the ribosomes, or code to the ribosomes, that the green needs to be put before purple or whatever. So the amino acids are put in the exact sequence based on the base sequences of the mRNA. So because of that, we say that the mRNA has a specific sequence of bases that code for a specific sequence of amino acids. Why we mention specific? Because I hear mention that the not all of them or not all the mRNA are going to code in the same way. It depends on the base sequences in them. So then the ribosomes are going to assemble the amino acids into polypeptide chains. They're going to be like long chains, which are then going to fold to form a complex 3D shape of an amino acid. It's going to be like this. Like this. And that way. Like somehow in a shape in which is 3D. Okay? That was about this question. Part III, DNA controls cell function by controlling the production of proteins. See two types of cell membrane proteins. Cell membrane. They're cell membrane proteins. I can say that they're protein carriers. And there are receptors.